So, good afternoon, everybody. We are going to start the last session of BSB 2020. We are going to have now the keynote, the last keynote, Professor Stoye. But before that, we are going to announce the best paper of BSB 2020. So, João, you can talk about the process of choosing the best paper, and then we're going to announce the best paper, okay?
Okay, thank you, Daniel. Welcome everyone to this afternoon's and last session of BSB 2020. This year we have the Best Paper Award. I will briefly explain how we arrived at our winner. So usually when you have a conference with the people actually present, you set up a committee, and the committee checks all papers that have been submitted, watches the presentations, and then makes the choice. This year, because we have an online conference, we had to follow a much simpler procedure. And the procedure was as follows. The full papers were submitted through the Easy Chair system. And in the Easy Chair system, the reviewers, they will score the papers that they have received for review. And the result of this process is a ranked list of papers. And the rank is based on the average of reviewers' scores. And you can weight the scores by reviewers' confidence in their reviews or without that weight. So it turned out that when I was the one who supervised this whole process, when all reviewers were done, and when I checked the ranked list of papers, there was a clear winner. One paper which had the best score, both in the weighted and the unweighted version. And the second best was a few decimal points below that. So there was no question that there was a tie. And so that was I thought that was the best way to choose the Best Paper Award, because it was totally independent of anything. So reviewers were not told beforehand that they had to choose a best paper. So it was entirely based on the rankings that came out from the Easy Chair system. So based on that, I will now announce what the best paper is. And so the winner is, please go ahead, Danielle. It's this paper, COVID-19 X-ray Diagnostic with Deep Neural Networks, whose authors were Gabriel Oliveira, Rafael Padilha, André Dorti, Luis Sereda, Luis Miyazaki, Mauricio Lopes, and Zanoni Diaz. So congratulations to all these authors for this award. And I would like to ask whether any of the authors are actually here attending this last session. Let me see. So please either announce yourself in the chat or raise your hand, or we wait just 1 or 2 minutes to see if anybody, if any of you is present. And if that's not the case, let's see here. I can see. I can see the list of people present. So it looks like, from what I can see here, none of the authors is actually present. And if that's the case, that's too bad, but we will send the certificate anyway to the corresponding author of this paper, who is Rafael Padilha. And with that, we'll move on to the next, to the presentation of our invited speaker, which I now introduce. So I'm very honored and happy to introduce you to Dr. Jens Stoye. He is a professor of genome informatics at Bielefeld University in Germany. He has been working in the field of bioinformatics for over 20 years, having made numerous contributions both on the theoretical side and on practical aspects related to data analysis and computational tools. He is one of the authors of the highly influential concept of double cut and join distance, which is a widely used model in theoretical studies of comparative genomics. Jens has been to Brazil very often. He has numerous collaborative ties here, as well as some family ties. He has visited on several occasions Universidade Federal Fluminense, who is the university who's actually holding this event, even though it's online. And so we're very glad that Jens has accepted our invitation to give this talk. And Jens, I welcome you to today's session, and thank you for having accepted this invitation.
João, thank you very much. It's a pleasure to be here, although much more I would love to be in UFI, in Niterói or somewhere, São Paulo. Actually, a year ago, I was still in UFABC visiting, so near São Paulo, Santo André. So I've been around for a while and in various places of Brazil. I love the country. It's a pity that I have to speak this way, but still better than nothing, I guess. So I will share my screen now so that everybody can see my slides. And I guess I will start with my talk, which is has the title "Searching for Genomic Variants in Multiple Genomes at Once." And this is actually joint work with a couple of people, this time not from Brazil. Actually, Tizian Schulz, the first author, is my PhD student. Roland Wittler and Sven Rahman are colleagues here in Germany, and Faraz Hash is a colleague from Vancouver in Canada, where I also, from time to time, have some collaborations. So this is work that I did in my role as a faculty member of the Faculty of Technology here, Center for Biotechnology, and also the Bielefeld Institute for Bioinformatics Infrastructure. So we have various institutions here at Bielefeld University where we, as Jean said, work on theoretical or more applied aspects of bioinformatics. So what is the topic of this talk? I start with a short motivation. I guess it's about genome sequencing. So as we all know, sequencing is very cheap nowadays compared to maybe 20 or 30 years ago, and lots of data are collected. And it's in fact a tendency now that while in the beginning people were focusing on model organisms and like reference genomes, let's say, that they were sequencing the human genome or the mouse genome or the Drosophila genome, whereas nowadays the focus has shifted a lot in the direction of individual sequencing. And especially for the human genome, there are all these big projects like 1000 Genomes Project. UK Biobank is a super big project, other national projects in various countries of the world. US has a big project for individual sequencing. So they're interested to really sequence personalized genomes and in the number of thousands or even millions. Yeah, and then comes the task to bioinformatics. What can we do with this? Yeah, and sometimes it's a very simple task. It's just how do we store all this? Yeah, so here on the right, I've listed some challenges. It's just efficient storage. This is a big amount of data. These are terabytes or petabytes of data, and we have to store them and make them accessible quickly. Another question is efficient analysis. Of course, it's great to have all these individual genomes, but we need somehow the technology to analyze it somehow. And the third is effective analysis. So also, it's not obvious how does an analysis look like. Yeah, one point is, for example, that all these genomes are, it's written here on the left, partially redundant. So actually largely redundant. If I compare several human genomes, they are very, very similar. Now, there are only some variations here and there. So to analyze it, it's a big difference to do this type of as we call now pangenomics, where you have the complete genome of a species in the sense that you analyze all the individual genomes, compared to traditional comparative genomics, where you would compare genomes from reference genomes from different species. So what is an effective analysis? That is the question that we also have to answer here. What can we learn, and how can we learn it from the data? And yeah, this brings me to the topic of pangenomics, where we are working on we call it the sequence-based pangenome. Is essentially illustrated by this picture. We have a couple of genomes here. We have three: genome A, genome B, genome 3, and they are highly overlapping. Yeah, so highly overlapping means there are large identical areas and only some relatively small areas where they differ, and the individual genomes come about. And so how can we store? The first question is, how can we store such pangenomes without repeating or storing the same information many times, which is inefficient? And there comes various data structures have been proposed for this. And my favorite actually is the De Bruijn graph. In fact, the colored De Bruijn graph, which is a variation of it. The De Bruijn graphs have been around for a while in bioinformatics, very frequently used in genome assembly. But here we use them more or less to store our data in an efficient way. So now let's look at the example just to get an idea what's going on. We have, let's say we have three genomes. Genomes are very short here, just these little 3 strings on the left, and each one gets a color. So this is the colored De Bruijn graph. So red, green, and blue. And then we take them and cut them into little overlapping strings, k-mers. So k equals 3 here, so 3 mers, and each 3-mer that we get this way is a vertex in our graph. So we see all 3 start with TGC. So we have a vertex TGC, and the vertex has colors. And because the TGC appears in all 3 genomes, we have all 3 colors, red, green, and blue, for this first vertex. Then the next overlapping k-mer in the red sequence is GCG. GCG is here, and is only present in the red genome. So it's only red, this vertex. And when we continue, we get CGA is here, GAT, ATT, until TTA. In the end, they all merge. The green and the blue have the second k-mer in common, which is GCA. But then they also diverge, and we get this bubble structure with the green path and the blue path. And in the end, also, again, it merges into TTA. So this is the data structure. And you see in this picture, it's not so visible, but if we imagine that the sequences have big amounts in common, then we need to store much less sequence than if we would store all the genomes, all the sequences individually, because simply each identical k-mer is stored only once and not 3 times in this case for the 3 sequences. And I did not really talk about the edges. The edges between these vertices, they're not so important. Later we will see that we can completely ignore them. But for the moment, let's say whenever we have 2 k-mers or 2 vertices in the graph, like TGC and GCA, that overlap by k-1 letters. So all letters except 1, like the TGC, the GC of TGC, which is a suffix of 1 vertex, is at the same time a prefix of the other, and then we draw an edge if this overlap has length k-1. And that's exactly what we see in this graph. So this is the colored de Broglie graph, but in fact it's easy to see that this compacted version of it has exactly the same information. So I go back again. Here we see this path, non-branching path of 4 red vertices, and here it's just compacted into 1 vertex. Yeah, so yeah, as we see here, the vertices of unique paths are replaced by a single vertex. And the label of this vertex is then the sequence that you get by spelling out the path. And this is even more efficient than the previous data structure because we have less vertices to store, although the sequence content is the same. So this is a very nice data structure and very powerful for many use cases. And there are efficient implementations of it. And I will talk about implementation a bit later. Yeah, just to recap a little bit. So colored de Broglie graphs reduce our memory requirement. That's actually the second point here is interesting. We often don't need any data pre-processing. So when we do comparative genomics in the classical sense, we need the genomes. We need the assembled genomes. And we all know that assembly is a tedious procedure and not always very, very easy and automatically doable. Whereas here, these de Broglie graphs, and this, of course, is related to the fact that de Broglie graphs are used to do assembly. The de Broglie graphs give us the chance that we can just take original read sequences from the sequencing machine and throw them into the de Broglie graph. Because we can, if we have a completely assembled genome, we would give it a color, blue, and put it into the de Broglie graph as I showed before. But if we have, let's say, 10 million reads from some species or from some individual, yeah, we can also take these 10 million reads, cut them into k-mers, give all 10 million reads the same color, and throw them into the de Broglie graph. And this has essentially the same effect. It doesn't really matter whether we have assembled or unassembled data. We can just throw them in, and this will work pretty nicely. Another point is that the data is stored based on similarity. So if we are somewhere in the graph and we see, like here, we have some area that is in the green, red, and blue sequence, and then the neighboring will probably also will be close in sequence space because there are these overlapping k-1 mers. So we can walk along the sequence by walking along the graph. And finally, and this is one of the main points of this work here, we can do the analysis when we have our input data, or our, let's say, database in such a format. We can do some analysis very easily with all data sequences in parallel. Yeah, we don't need to compare to 10,000 individual sequences. We can just compare to one de Broglie graph, one colored de Broglie graph, and everything is done at once. So this is very efficient in practice, as we will see later also. Okay, so now the thing that we did, I mean, colored de Broglie graphs have been used already for maybe 5 years in this area, so that's nothing new. But until recently, there was no software. I mean, everybody knows BLAST, and this slide here is about BLAST. So there was no software that does a BLAST-like alignment procedure against a colored de Broglie graph. So these, I mean, BLAST traditionally, like the classical software that everybody knows, is for either DNA-DNA alignment or protein-protein or translated DNA against protein or protein against translated DNA. So all these flavors exist, but it always is against a sequence database. Yeah, it's a database of individual sequences stored in a kind of multiple FASTA format. Yeah, not a graph. And this is what we thought is a pity, because you lose all this efficiency from the redundant data, because you blast or you search against very similar sequences many times, whereas if you have them in the graph represented, you would search only once and get the redundancy as a speedup. And that's exactly what the work that I'm presenting here is about. So let's recap what are the three main steps of BLAST. So BLAST has these steps as indicated here in the bottom right. The first thing is BLAST finds these so-called BLAST hits, so short identical matches that are at least some length, I don't know, W maybe. These are the seeds of an seed extension algorithm. The second step is then the extension, where they develop this X-drop algorithm that says I have a seed, and from the seed I go to the left and right, and hopefully I get, although I allow for mismatches, no gaps, only mismatches, no insertions, deletions, but I can, after some mismatches, maybe get more matches and increase the score and increase the score. But once the score gets really low, much lower than some intermediate score that I have already seen, then I stop. Yeah, and this is the X-drop extension algorithm, which is very powerful in BLAST, and so is kind of the standard in this area. And the last, once we're able to do a good X-drop extension to the left and or to the right, then the last step is that this whole area will be recalculated, a new alignment will be calculated, now this time with gaps. So in the third step, also insertions and deletions can be accommodated, so that we get a complete alignment as output, as we know it from textbooks. Okay, that's the three steps of BLAST, and essentially when we phrase this as a computational problem, we would say, given a query sequence X and a sequence database capital Y, find all high-scoring local alignments between X and any sequence small y in Y. Above, that's also important, above a certain significance value. So there is some statistical aspect in it that considers how significant is a hit in this sense, and only the most significant ones will be returned along with their score. So we get an alignment and the alignment score, but often also annotated with an e-value, which is kind of the significance value that shows the smaller the e-value, the more significant is the hit. Okay, that's the situation for BLAST. And now we said, why can't we do essentially the same with the colored de Bruijn graph? Yeah, and the rest of the talk is essentially about this and how we had to adapt, because it's not always obvious. I mean, some parts are kind of clear, but some parts are really a bit tricky. And I will guide you through this development. So the idea is clear. Develop a method that queries a colored de Bruijn graph in a BLAST-like manner to find local alignments between a query sequence and the sequences stored in the graph. Yeah, so we do not repeat anything. We do one search against the whole graph, but because the graph contains all the sequences, we search against our complete redundant sequence database at once. And the tool that we developed, we call the Pangenome local alignment search tool, short BLAST with a P. And the problem statement is very clear. It's more or less the same as we have seen for BLAST. We have given a query sequence X, and also a set of sequences capital Y, but this time represented as a color de Broglie graph. And then we want to find all highest scoring local alignments between X and any small y in capital Y above a certain significance value and return them along with their score. So same situation, same problem, except that the database is not a multiple FASTA file with many flat sequences next to each other, but one color de Broglie graph, so that we have to somehow handle this different data structure in our search strategy. Oops. But the search strategy, apart from that the data structure is different, we really try to copy BLAST as good as possible. So we also want to do a seed detection first, seed extension, and a gapped alignment recalculation. Except that the input now, the input database, is a graph that could look like this, and I will guide you through this example in step by step, just to give you an idea also what can be a bit tricky here. So let's say this is the graph. In fact, for the moment, we forget all the colors. So let's just search for the presence or absence of local alignments. The colors will, I will say a few words a bit later, how the colors can be added again. We will also forget about the edges, because in fact, the edges are determined by the vertices. Yeah? As I defined earlier, when we have two vertices, we can always say whether there should be an edge between them or not. Let's look here. We have the vertex A, CC, TAA, and TAAC. Here the value for K is 4. So there's an edge if and only if there is an overlap of the two sequences of length 3, and there is TAA as a suffix here equals TAA as a prefix here. So this is an edge, and we can reconstruct the fact that there should be an edge just from the labels of the vertices. So we don't really need to store the edges, as long as we have a way to access the labels of our vertices very quickly. And that is possible if we use some data structure, let's see, no, well, some indexing data structure for the graph. And here is an example. So when we are looking for 3 mares in this graph, for example, then we would can do one table that is essentially all 3 mares from AAA down to TTT, and each of them is a pointer into another table, and the other table contains all of the pointers into the graph where the corresponding 3 mare occurs. So if we're looking for AAA, then we know the first entry here tells us where is an AAA, and it's vertex 10 with an offset of 0. So we search for vertex 10, it's here, offset 0, AAA works. So the next AAC, there's a 3 here, which means after the 1, but up to position 3 in this table, we will find all occurrences of AAC in our graph. So at vertex 7, offset 0, and vertex 11, offset 0, we will find AAC. Here is 11, offset 0, here's 7, offset 0, and so on. So these two tables, which are maybe a bit big, but very easy to handle, they contain, they give us very fast access to all K-mares or K-1 mares in the graph, which is very, very practical. Okay, and now let's go to the seed detection algorithm. So now we have a query down here, X, and let's start somewhere in the middle here. And we already find, using this table, we find a seed of length 5 here, TAACG. This appears on the vertices 8, 9, and 11 as a path in the graph without any mismatch. And this is a seed. This is a seed. There are many more seeds in this example, but let's focus on this one. Once we have found a seed and we cannot extend it without introducing mismatches to the left or to the right, then we come, whoops, okay, possible in the same, yeah, essentially what I just said. We use the pre-calculated index of the graph. And it's in fact very similar to what BLAST also does. And then we come to the second step, the seed extension. So here we have our path. We have the seed extension. We extend to the right. The next in our query is a G, but there is no G here. So we have to add a mismatch. And oops. And the mismatch is what is indicated here with a little minus sign. That means we are like, yeah, we have one mismatch. But then if we continue, we find a match, we get a G. And we continue maybe along this edge here. I mean, we have to go in two ways because the graph is branching. Let's go here first. We find another G, so we have a second plus. So now we already have a better score than before. If we say that a minus is a score of -1, a plus is a score of +1, then we are already better than we were in the beginning. So then next, if we continue, we cannot continue. We get only CCC, which is does not match at all. So we would stop here with this X drop extension. But we have another branch. So we go back. Oops. Huh? Yeah, because as I said, there is nothing to extend here. There's another branch that's this edge going up. So here the next letter after the T, TG, after the G is a T, and then comes another T, so we have minus, but then we have a plus, we have an A that is a match with the query, we have a G that is a match with the query, we have a T that's a match with the query, and this looks very good. And we can even extend into the next vertex, and we have an A, but that's a mismatch, so we would stop here. So we have this as a second hit, and as a second hit, and this second hit is even better. So we see that we have to explore the graph in a kind of depth first way, which can be a bit tedious, but or expensive, as I'm saying here, because the graph sequence is ambiguous, so we have to go in various directions. But most of the cases, it is not a big problem. Most of the cases, the graph is not very, very tangled, and we have a couple of branches to walk. But if there is no good similarity, the X drop stops fairly soon, and we don't have to traverse the graph too much. Only in very rare cases, but sometimes this happens, we have what we call the endless extension problem. So that sometimes the graph is really very tangled and we get many collapsed regions, and they are very, very similar. And if then they are also similar to the query sequence, we might get many short loops and vertices with short, and if we have the high branching factor, we have in fact to traverse many paths, and that's the if that is the case, we have to stop this somehow brute force. So there we implemented a hard break where we say if the X drop algorithm is unable to end the extensions, for example, if after 10,000 steps it still has open branches to follow, then we just stop it. And most of the cases, this is no problem, because this is a very tangled area of the graph that where many paths have very similar score, and it doesn't really matter which one is the one best path. If we have found one that's pretty good, that's enough. So this is the little problem that we had here, which is not a problem in BLAST, but we could, with this heuristic, we could somehow handle it. The third step then is the gap alignment that I said, and this is essentially done in the same way as it's done in BLAST. So we do a banded alignment of the area in the graph that we have found. So we have found an area in the graph. We take the neighboring sequences in the query. We try to extend it. We add gaps here and there. So we see that there can be insertions or deletions. And this gives us, in the end, the output of our algorithm. Of course, this is even more expensive than the second step, because here we have to traverse the graph in various directions, where we even have to consider insertions and deletions, gaps. But fortunately, this third step happens only very few cases. Yeah, so we will not do this too often, and therefore it's also computationally very feasible. Okay, that ends the description of the algorithm. Two things that I promised I would mention is what about the colors in the graph, in the color tiptoeing graph? So how can we consider the genomes that were the individual genomes that were the building blocks of the graph? And there we have two ways to handle this. One is a quorum parameter that we say, for example, one might be interested in finding hits that are present in 90% of all genomes in my graph. Yeah, and if the graph has, for example, 100 colors, 100 genomes in it, then we would restrict all the search that I just described only to those vertices that have at least 90 different colors, 90%. Yeah, so this is a quorum, this is a parameter, which doesn't change the algorithm much, but can, as we will see later, can give us some flexibility how we organize our search. And another one is even more explicit, that we explicitly enter a search color set. So we say we want to find hits, but only in this subset of the graph. Yeah, and later I will present some results, or mention some results from the 1000 Genomes project. And there we, for example, can easily restrict the search to only those genomes from African origin. Yeah, we just say, okay, here are the 350 African genomes, and we only want to search in that. And that's the same algorithm, just that when we do the counting and in the seed detection, we would only check that the vertices have at least one African genome in it. Otherwise, we would stop the search. So these two little parameters don't change the algorithm much, but allow us to do all kinds of interesting queries to the graph as a whole and to subsets of it. All right, another aspect I have already mentioned is alignment statistics. Yeah, so BLAST, if you remember the old days, there were essentially two fast sequence analysis tools. There was FASTA and there was BLAST. But BLAST had the better statistics, and therefore, after some point, people would always prefer to use BLAST than FASTA. And so we also thought that we should understand the alignment statistics here a bit better. Oh no, let's first look at the BLAST statistics. I mean, BLAST is the so-called Cullen-Altschul statistics, where we have an e-value and a p-value, and the e-value is essentially given by this formula, where the input parameters are the query length and the database size. And we thought it would be very nice if we also can somehow, just by some structural parameters of our graph, develop a statistical measure for significance of hits of a query against a pangenome graph. This was actually not so obvious. So we had to take some assumptions. One assumption, and that speaks against the similarity of our statistics to the BLAST statistics, because we have, usually in all these statistics, people assume independence. So independence of the data in the database, for example. But this is explicitly not given for us, because we know, and that's the whole point, that our genomes, our input data is highly redundant, so highly similar. So the randomness assumption is definitely not fulfilled here. On the other hand, our graphs have this branchings, and we can create some sequences in the graph that are never input sequences of the input genomes. Yeah, so that speaks against the first point, or in favor of randomness, because we can create, even though maybe we have only 10 input genomes, we can create maybe hundreds or thousands of different sequences in the graph by combining them at the branch points. So that brings again some randomness into the game. Well, the main assumption that we do is that the maximum scores, like in the Karlin-Altschul statistic, follow an exponential distribution. But this is only an assumption that we have to verify by experiments, and I will show this experiment in a second. And so the two parameters inside the formula, this lambda and c, they depend on the sequence relatedness and diversity within the pangenome. So this is our assumption where we believe it's true somehow, motivated by these first 2 points, but the details are unknown, and I guess there needs to be a real statistics person to look at this and go deeper in this. How much random and non-random are our sequences against which we compare? Anyway, we did some experiments, so we estimated these values of lambda and c for a given data set. We could not directly find properties of the graph where we could run this, but for a given data set, one can run estimates. The simplest way is some naive sampling, that we just produce random sequences, align them to the graph, and then see how is the score distribution. But of course, by random sequences, we get often very low scoring alignments, and we need a lot of sampling, and this takes very long. Unfortunately, there's another way that's called importance sampling, where we can somehow not create random sequences, but random query sequences, but create sequences that are somehow modified so that they are specially handy for and produce especially similar to the database sequences, and therefore produce higher scores. And maybe I skipped this here a little bit, but the idea is to construct a Markov chain, and the Markov chain produces query sequences that, if you compare them to the graph, are still kind of random, but with a very high probability produce higher scores than purely random picked sequences. And given that, then we can estimate with some intermediate steps these two parameters, lambda and c, of this Karlin-Altschul statistics. And we did all these simulations, and here we also see that they worked very well. So first we did the naive sampling. These are the x's here, more in the low score area, and we see that there is, in fact, when we see a straight line in this plot here, this means that there is we have good parameters for lambda and c, and we see that after the scores we reach the tail distribution, so the lower end of, or the higher end of high scores and low probabilities, where we are in fact looking for significant hits. Then we see a good straight line, initially the axis, but from the axis then we use the importance sampling, and with the importance sampling we can extend this line very far to the right, and we see that that's really straight. We have to be careful. We need two statistics. We need one statistics for the ungapped alignments after the second step of our algorithm, and then we need another statistic for the gapped alignments after the third step. And we see that the two are also don't have exactly the same parameters. So the one curve, the gray curve, is a bit steeper, and the black curve for the gapped alignment is a bit lower, flatter, which is also expected. Here on the right, we see that with just the score distribution of the queries for our sampling, and with the blue and purple is the importance sampling. So we see that we can get much higher scores with the importance sampling than with the naive sampling. As expected, but it was not so easy to do this, but now we're very happy with the results. Okay, so this was the statistics, and now we come to the results with the method itself. So, of course, when we compare BLAST, the first thing is we want to compare to BLAST, but of course, as I said initially, we are in a bit different situation. So BLAST does a different job. It compares the query to each database sequence, whereas we compare the query to each sequence that's stored in the graph. And in fact, and this is an interesting point, by doing this, we also compare the graph sequences among each other. Because if we get a hit with some query sequence of 5 sequences in the graph, we also learn that these 5 sequences in the graph, at least in this area, have a strong sequence similarity. Yeah, so that's why we can not only search many sequences at the same time, but we can also compare all of the sequences to each other. And this is, in fact, often what we see in a BLAST type or in a standard sequence analysis procedure, that people need a lot of post-processing. So they do a query, they get some hits from different genomes maybe, and then they have to compare all these hits to each other in a quadratic fashion. Whereas we don't need to do this because we get it automatically from the graph for free. So that's pretty nice. Also about post-processing analysis that often comes after a BLAST search. Okay, so let's do just a pure runtime comparison to some other tools. So we compare to BLAST, which are the black circles, to BLAT, which is the BLAST-like alignment search tool, which in fact is even slower. So here we see running times for different number of genomes. These are Salmonella genomes. We have compared to MMSEC2 and uBLAST, which are two more modern sequence analysis tools, and the open circles are our tool. So we see that for small values, we are somewhere in the middle, but when we go to high number of genomes, we see that well, I don't know about the curve of BLAST, the black circles, but all others kind of scale linearly, whereas ours, I think, quite clearly scales sublinearly. So you can add more and more genomes, and still it's not that proportional with the number of genomes the running time increases. In fact, it gets, the increase per genome gets lower and lower as we go here to the right. Yeah, and we could actually do much more than 5,000 genomes here, but I think we didn't have the data, and some of the other methods. Here you see this uBLAST. uBLAST is super slow, so we could not compare it at all. But another aspect is also memory usage. So here we see that another very fast tool is this MMSEC2. But there we also see that if we look at the memory requirement, this is by far the worst. So MMSEC2 uses lots of memory compared to the other tools. And in fact, our BLAST uses by far the lowest amount of memory here, also for the big ones. I mean, memory is for many of the tools not a big issue, but we are the best anyway, by a factor of 2 more or less, which is quite nice. But it's no surprise because the memory is essentially the graph, which is employing all this redundancy in the data. That's one of the basic ideas. Okay, but what about the hits? What do we find? And this is a very complicated table, and I don't like to show it in this way, but I had no idea how to present these numbers in a graphical way. What we see here on the left, first, let's just focus on this column. For the 5 tools, the number of results. And this is a somehow interesting number, and again repeats what the tools do. For example, we get only 7,500. I mean, this is again the same. Let's go back. The same 5,000 Salmonella assemblies, and we get 7,500 hits. So that means we have maybe 1.5 hits per genome and the query is a sequence from the Salmonella reference genome. So these are Salmonella typhimurium, and the query is Salmonella reference. So per query we get 7,500, where there are 5,000 genomes. So but BLAST, for example, gets 1.2 million hits. So how can we compare this? But of course it is because we compare only against the graph. So our hit, when we have a result hit, it could be a hit where we see out of the 5,000 genomes, we get maybe 3,000 in one hit. Yeah, because there might be vertices involved that have 3,000 colors. Whereas BLAST, each hit is only against one genome and against one genome and against one genome. That's why all the other tools have much higher numbers than what we get. But this is somehow not a bad property. I find it, I really like this result because we can focus on the essential. Yeah, I mean, these 1.2 million hits of BLAST, or even 4.8 million hits of uBLAST, you have to post-process. Yeah, there are just too many. Whereas, and you have to see which of them appear in many of the genomes, which are singletons, and so on. Whereas we get all this for free when we do the Pangenome approach. So that's one. And the other, this table here essentially shows us how many hits does one tool find that another tool does not find. And for that, we say that 2 hits or 2 alignments match if they overlap by at least 90% of the shorter alignment. So, and then we count how many alignments one tool finds that another tool does not find. And we see that the numbers are generally very small if we ignore uBLAST. I mean, uBLAST somehow has a problem. They find too much. Yeah, they find much too much. So the numbers down here are too big. But here we see that for example, BLAST finds only 290 of these 1.2 million hits. Only 290 of them we do not find with BLAST, with one of our 7,500 BLAST hits. So almost nothing. And here blood even less, only one. They find 450,000, and we also find all of them, and so on. So essentially, I guess this table is very good news, that we find more or less the same thing that the other tools also find, ignoring uBLAST, and but in a much more compact way that can be very, very useful. Okay, there are some tools that we have not included in our analysis for various reasons, but doesn't matter. Okay, now I have 2 short use cases, and then I'm done. What we can do with this kind of analysis. So one is this Vibrio cholera bacterium that has some strains that are pathogenic and some are not, and there is this VPI1 region that has 19 genes associated with it that are somehow considered this the causes this pathogenicity. And we searched for these 19 genes as one query sequence in 21 Vibrio genomes. And there are some genomes where we find almost all of them. That's on the left side plotted. You see lots of purple. And there are other genomes where we find almost nothing, none of these 19 genes. And we can do this with one query of these 19 genes concatenated against the Pangenome graph. Then when we look at the annotation of these genomes, these on the left are exactly the pandemic genomes, whereas the ones on the right are environmental genomes that are not pathogenic. So, of course, somebody has to tell me which are these 19 genes and that I should look for them. But once, maybe if somebody gives me an unclassified Vibrio cholera genome, I can quickly find out, adding it to the Pangenome graph, and find out, does it fit into the one or into the other group. And the second use case is about the human. So we can also construct the pangenome for the human genome. We could not do it in our analysis because the other tools could not do this, but we have done it. Here we have done it for 2 chromosomes, chromosome 2 and chromosome 15. Chromosome 2 is already pretty big. For 2,500 human genomes from the 1,000 Genomes project. And we were looking for some polymorphism in the human genome. It's this SNP RS1426654 that has something to do with skin pigmentation. And it's known that the West Eurasian ancestry has a different allele from the rest of the world. And it is somewhere on chromosome 15. So we used the pangenome of chromosome 15. We were searching for this region. And when we search for this region with no quorum, so just give me everything, we will get 2 hits. We will get a perfect match, like in the reference genome, and we get another match that has a single, which is a variant that has a single mismatch at 1 position. When we go to 99% quorum in all European samples, so this is now this quorum thing that I mentioned earlier, that we can restrict our search using the same graph, but just with 1 parameter restrict the search to some subset of the genomes. We get only the reference allele. So we get exactly only hits in one of the groups and not the other one. So all these kind of analysis can be done very, very quickly. Let's say, yeah, I guess it's quickly. So the experiment with the chromosome, oh no, now we have chromosome 2. So it's the bigger, one of the biggest human chromosomes. And here we can do a search in 305 minutes and use 24 gigabytes of memory on a single processor. Whereas this MMSEX2 software uses much more memory and is essentially the same speed. So we are, except that they also have terabytes of disk space that they use. So this MMSEX is fast, but has a big memory problem, as I said before. Okay, I guess this is the end of the story. I guess I told you that with increasing amounts of DNA, the sequenced the projects are going more and more in this pangenomic area, both in bacterial and in human or eukaryotes. In fact, we can also, when we look at viral, just because everybody talks about viruses these days, some people also say that viral genomes form a pangenome, but in fact, there I would say we have to be a bit careful because viral sequences have very different characteristics, and our data structures are not optimized for that. We have shown by our software that it is possible in practice to run this BLAST-like algorithm on a compacted de Bruijn graph. And yeah, I think the main point is that essentially we can, by this search, we can do in parallel what other software that is searching against classical sequence databases has to do sequentially. And in many cases, we can even avoid complicated post-processing. Okay, this is the end. I thank you very much for your attention, and I'm looking forward to answer questions. And the manuscript is on bioRxiv for this, and we hope there will be a publication soon.
Thank you, Jens, for a very nice talk. Very interesting topic, very interesting tool. And we'll now have time for questions. If people have questions, I ask them to write them on the chat area. Jens, you may want to stop sharing your screen so that we can.
Yes.
Okay. So let me start by asking a question myself. You did not say anything about the time to build the color De Bruijn graph, as well as the time that, or the effort that may be required to continually update it, because once you build a pangenome graph, it's of course the case that you know, depending on the organism that you're working on, every week there will be new genomes. So could you talk a little bit about that?
Yes, I mean, I did not talk about that because it's not part of our work, but I should have mentioned, I should have given credit to actually a former PhD student of mine, Guillaume Holle, who's now in Iceland, and they're developing this software called Bifrost, Bifrost, which is not only a Nordic god, but it's also a software for creating colored De Bruijn graphs. And that is what we use essentially out of the box. And all our analysis, I mean, we add this index that I presented, the index to get fast access to the K-mirror, to the seeds of the search. So in fact, your first question is twofold. The first part is how long does it take to build the graph? And the second is, which is another pre-processing step, how long does it take to build this in our index? And I have to say the graph is very fast. Well, no, the graph creation is not so fast. I think it is, well, I don't have numbers here, but it takes for a bacterial pangenome, it takes some hours. But the graph can be stored on disk. And once it's on disk, it can be loaded very fast. Yeah. One problem is that our index is, and the graph is pretty small. So that's also nice. The Bifrost, they use minimizers and some really fancy algorithmic tricks that they have to use, but it brings the graph to a very low memory footprint. Actually, our index that we add on top of the graph uses maybe 10 times as much memory as the Bifrost graph underlying. That's a bit unfortunate, but so far we have no idea how to make our representation more compact. On the other hand, it's still, memory is not an issue. Time is an issue in the beginning, in the initial creation, yes, but that is hopefully done only once. But this brings me to your second question, the dynamic data structure. And indeed, Bifrost is a dynamic data structure. So Bifrost you can add and remove genomes as you wish, and everything is handled nicely and smoothly. That's not our credit, but they did this really in a very good way. Unfortunately, our index is not dynamic. So the answer is partial yes, partial no. So the Bifrost, you can add genomes, remove genomes, and so on. But as soon as you want to do an analysis of our type, you have to create our index for the given genome. And when you change the graph, you have to rebuild the index from scratch. That's a bit unfortunate, but also there, you see, we have not really optimized our index so far. Maybe one could also think about making it dynamic in the future.
Okay. So there is one question in the chat. Probably you can read it yourself.
Yes, so I just read it and try to pause it while reading. Great presentation and good to know about BLAST. Thank you. Just wondering, back to the initial part concerning the size of the De Bruijn graph in your experiments, do them all fit in RAM? Yes. I mean, it's a bit overlapping to Jean's question. I think the De Bruijn graph is really not the problem. The De Bruijn graph, even of big human genomes, fits in RAM. Because of course, it depends on the size of the genomes that represent, but even more on the redundancy of the genomes. So as far as I know, they have done, I mean, it depends on the machine that you have, but on typical machines now with maybe 10 gigabytes of main memory, you can store a human genome of a couple of hundred, pangenome of a couple of hundred human genomes. So this is a very compact representation. For BLAST, not. I mean, as I said, our index is a bit more greedy there for memory. There, I would prefer to do it chromosome-wise, for example. So each chromosome you would fit in memory, but not the whole index for all human chromosomes at once. But then you can parallelize easily and it will work again. Yeah, so memory, at least for the underlying data structure, is not a big deal. Very nice.
Okay. I actually have another question. The examples you gave were from the human genome and from Salmonella typhimurion.
Yeah.
And in principle, in theory, you could extend your data structure to include more general categories. So, for example, the pangenome of all bacteria within a certain genus. Yes. And you could then go on and on and on, and at least in theory, you could build a pangenome graph of all bacteria. So yes. Probably that's not a good idea.
Yes.
I can imagine a few reasons why it wouldn't be a good idea, but my question is whether you have a feeling as to the right scope for this data structure.
Well, you're asking exactly the right question at the right time. And now I have to promote another former student of mine who's actually a co-author of this paper, Roland Wittler, because he uses pangenomics for exactly that question. He has had a paper last year about a software that is called SANS, and it's using pangenomics or using color T-Breuil graphs, the same Bifrost implementation that we also use here, to do phylogenetic analysis. And so like organizing, yeah, phyla of the bacterial kingdom. And that's what he's doing. And there he's quite successful. So he's doing now metagenomics with exactly his tool. I mean, it's only loosely related to the BLAST search here, because he uses the pangenome, looks for paths in the pangenome that have common colors, and then puts them, creates splits, if you know splits theory and in phylogenetics. So you create some taxa against some other taxa, all these splits, and from all these splits together, you can puzzle a phylogenetic tree. And he does this right now for subtrees of the bacterial kingdom. And he had the same idea. What happens if I use sample the whole space of bacteria, and he gets nothing good, because the space is a big mixture, too diverse. But now I'm not aware. I have not seen his talk yet, because he's ongoing in this project. But I know that he is now focusing with some metagenomics people here in Bielefeld together on some bacterial families or phyla. I don't know, some definitely above species level, but below kingdom. And there he's quite successful, actually. And he said, he told me last week in our regular meeting that he now has a setting where he's pretty happy. And then you you have the given taxonomic tree of the bacterial area that you're looking at, and then you have unknown taxa, like in metagenomic taxonomy, throw them into the same graph, and then you see where do they cluster together with which of the known phyla, and then you can hopefully and the good thing is it's completely alignment free, so it's fast, it's based on k-mers, so very quickly you get this initial draft overview where your new taxa would sit inside the bigger tree. So it's an approach that I'm not really involved in this project, but here in the group we are working on this, and I hope to see results soon.
Okay, excellent. Well, let's see if we have any more questions. Well, doesn't look like we do. If there are no more questions, then I will thank you, Jens, once again for having accepted this invitation. And it was a great talk, and we will now proceed with the closing part of the whole event. And so it's over to you, Daniel. Thank you.
Thank you, João. Before finishing this session and the event, I'd like to start thanking all people involved in the BSB organization. I'd like to thank you, Professor João Setúbal, Professor Valdeir Mendes, Professor Sérgio Lifshitz, Professor Raquel Minardi, and Luis Coada. Without you, it would be impossible to organize this event. I'd like also to thank you, all the students that supported us during this week, Maria Luisa, Carlos Gracioli, and Lenora. I'd like also to thank the Brazilian Computer Society for supporting this event and FAPERJ for sponsoring it. And of course, I'd like to thank all keynote speakers for their excellent talks, and for all of you that are attending the session, and attending all the previous sessions. We are all very worried about this online version of BSB, but I think this was a very good event, as we discussed and as we saw during this week. The Special Interest Group of Computational Biology of the Brazilian Computer Society has already started working on the next year BSB, so we are going to have a BSB next year, so prepare your works, prepare your papers for submit for next year. I hope to see you, all of you, next year in the BSB 2021 in person, not a virtual event. So before finishing this session, I'd like to thank you, all of you, for your great effort to make the BSB 2020 real. Thank you very much.
Thank you, Daniel.